Good morning, everybody. Thanks for joining this Grand Rounds. It's my pleasure to introduce Kevin Lin.
Recording in progress.
Thanks, Amy. So Kevin actually graduated from UC Berkeley with a Bachelor's of Science in Bioengineering back in 2013. So Kevin actually developed an interest in bioinformatics research after his undergraduate. He was working in a melanoma research lab at the University of California, San Francisco. Worked there for about 3 years. Doing bioinformatics and computational-based research in cancer before joining the University of Minnesota's medical scientist training program in 2016. Kevin's done his PhD in the bioinformatics and computational biology program, or the BICB program, for those of you that are in the know. And he was actually co-mentored by both Chad Myers from the computational bioinformatics side and Anya Bielinsky, thinking about the DNA repair aspects, cancer biology, and response to targeted pharmacologic agents, which is kind of what a lot of his talk will be focused on today. During his PhD years, Kevin also did his clinical continuity pathway with myself. So Kevin spent a lot of time with us in the molecular pathology laboratory, learning the ins and outs of how we do our jobs and how we interpret our predominantly NGS cancer cases. He worked with Dr. Sophia Yohe and Jeff Yang as well. In addition to molecular pathology, Kevin's had a chance to spend some time with Andy Johnson, Molly Klein, and kind of get a sense of what pathology is across the spectrum in a couple of different disciplines. So he's quite interested in thinking about pathology as a future career after he finishes MD, PhD. He recently published a STAR protocols paper on the work that he'll be presenting today, and as well, he's also co-authored a case report with myself and one of our former residents on HER2 del16 expressing colorectal cancer and how that affects outcomes and response to trastuzumab. So with that, Kevin, I'll let you take it away, and you can talk to us about your scalable chemical CRISPR-Cas9 screen and how we can predict drug mode of action from that. So thank you very much for being here. Appreciate it.
Yeah, all right. Thanks, Andy. Thank you for the kind introduction, and thank you all for having me here and having the opportunity to present my work. As Andy mentioned, I'll be talking about this scalable chemical screen that we've been working on for the past 4 or 5 years. Before I begin, I just wanted to acknowledge my doctoral advisors. So this work that you're about to see is a collaboration between both of these labs. Chad Myers, who is in the BICB in the Department of Computer Science and Engineering, his work mostly focuses on functional genomics and network biology. And on the right, Anya Bielinsky, many of you have known, is recently moved away to UVA as the chair of the biochemistry department there. She focuses on DNA repair and replication. And so one thing that I do want you all to take away from this presentation is that this is an integration between bioinformatics, computer science, and DNA repair and replication. And in terms of how it applies to clinical therapeutics. So with that brief introduction, I just want to give an outline of the presentation. First, I'll start with the clinical motivation for the work, then go into spend some time on the background. I suspect that the work that I'll be presenting today will be unfamiliar to many of this crowd. So I'll spend some time on that background. And then go into describing the scalable screen platform and its application to drug discovery. And I'll end with some final thoughts and summaries. So in terms of the clinical motivation, so what my project really is after in terms of a long-term goal is a more efficient approach to drug discovery. Many of you are familiar with the drug discovery pipeline, where we spend a lot of time in the basic research and drug discovery side of things. And there's basically a holdup here in terms of a bottleneck in terms of discovering new medicines. And so we're also familiar with the later stages of clinical trials and FDA review. It takes 15 to 20 years for a drug to hit the market. What I'm going to focus on with my work is really addressing that bottleneck upstream in this drug discovery area. And so I'd like to start with just talking about this paradigm shift in drug discovery. So a lot of us are familiar with the target-centric approach. So what do I mean by that? It means that we have, say, a protein target of interest. And we build this in vitro functional assay around that protein target for the purposes of screening this against a large compound library in the format of a high throughput screen. And the hope is that we will get some compound hits. And we take this assay, we'll look at these hits and hopefully find something that has an on-target effect, avoid the off-targets. Now, this approach is inherently limited in its scalability. And what I mean by that is that you have to develop that biochemical assay for each protein target of interest. And you can imagine with so many proteins out there that scaling this up gets to be a considerable amount of work and is in some ways inefficient. So the central question here is, can we make the drug discovery process more efficient? And so one possible way to do that is to flip that paradigm on its head. That is, instead of going from a target-centric point of view, you go from a compound-centric point of view. And so the idea here is that you have a method that looks at a compound and understand the biological processes or the target space that it perturbs. And so if you have this method that is compound-centric, you can imagine scaling that up to large compound collections where you have hundreds of thousands or millions of compounds. And you can really understand a lot of these compounds and their predicted mode of actions at scale. And so that's really what my thesis work is trying to get at is, can we develop an approach for that compound-centric drug discovery? So before I go into that, I want to go into some background of genetic and chemical genetic interactions. I'll first start with genetic interactions, which is defined as the combination of mutations that yield an unexpected phenotype. So what I mean by that is that basically when you look at a mutant in gene A versus a mutant in gene B, looking at the combination of the two and seeing its effect on a phenotype such as fitness, which is one of the most quantifiable phenotypes. In the clinical world, we're very familiar with the most severe negative type of genetic interaction. That would be the synthetic lethal interactions where, knocking out gene A and B individually has some sort of a phenotype, a fitness defect. But what happens is that when you combine them and they're both pointing at some essential function of the cell, you are essentially killing the cell where this phenotype goes to zero. And so, we're very familiar with something like PARP inhibitors in the context of BRCA mutant cancers. And that is an example of synthetic lethality. What I do want you to appreciate, though, is that that's just the most extreme example of these genetic interactions. And so we can really quantify these GIs on a spectrum. And so instead of it going to zero, you can, and I'll take this example here, where under some multiplicative null model, you would expect the combination of knocking out gene A and gene B to have some multiplied effect. For a negative interaction, that's anything really to the left of this gray bar. And so we call these synthetic sick interactions, but basically anything to the left of this bar, a lower than 0.4 effect, we quantify as a negative interaction. And then conversely, on the other side, we have positive interactions, where again, each mutant by itself has a certain fitness defect, and its combination is expected to hit, say, 0.25 here. But if you go above that, your fitness is better than expected. That's a positive genetic interaction. And so this scenario on the right occurs where you have A and B, say, in the same pathway or part of the same protein complex, where knocking one out, say, gene A, and then knocking out B will not worsen the effect. So we have these negative and positive genetic interactions. So how is this biologically meaningful? Well, if you put this in the context of fingerprint profiles, we can begin to see where that meaning comes out. And so what I mean by that is on the right here, I'm detailing again an example of synthetic lethality, or a very severe negative interaction up here. And what would be interesting is, say, if we have a genome-wide deletion library, and that's what you see on the column side, meaning that we knock out every gene in the genome of interest, and then we pair that with another gene knockout, such that each of these squares is interrogating a double mutant knockout. We can then form this matrix where for every query gene knockout, so for take gene A and knocking that out, you interrogate that against the genome-wide deletion library, and you get these quantifiable GIs. Blue here representing the negative side, and yellow representing the positive side. And so, okay, we get these genetic interactions and these genetic interaction profiles that are essentially fingerprints or signatures for a given gene A knockout. And we can do this for, across the entire gene or the genome. And so this work has been done by our collaborators at the University of Toronto. Over in Charlie Boone and Brenda Andrews' lab. They were at the forefront of this automated yeast genetics work. And so the idea here is that we can, if you think back about that matrix of genetic interactions, on these plates, we can actually automate interrogating those double mutant knockouts. And so this work, published back in 2001, we're able to do this across the entire yeast genome using this what's called the SGA, or synthetic gene analysis genetic array method. We can automate these with robotics and really interrogate all 5,400 genes in the yeast genome and do these pairwise double mutants. And then in assessing the fitness defect, you can basically then quantify these genetic interactions. And so back to how are these profiles biologically meaningful? When you create that matrix, what's really interesting is that you have certain rows that look similar to each other. So that's what you see here with gene A, B, and C. There's one cluster here, and then D, E, F, G is another cluster here. And what's really interesting is that the similarity of those profiles or those signatures tells you something about how correlated those genes are in terms of their biological functions. And so this is where the Myers Lab over here at the University of Minnesota provided the computational support for quantifying this and building a profile similarity network. And so when you really put this into a network graph, the nodes are each gene, and the edges here are referring to the strength of the correlation of the profiles between, say, A and B. What you see is that, or what you can build is this very pretty network map of genetic interactions. And so this has been done in yeast and published in Science in 2016, where we've built nearly every possible double mutant in the yeast, and that comes out to be 23 million of these double mutants. And we can build this functional map. And what you see here is that this map sort of self-organizes into these biological process bins. That is, we can, taking the 5,400 genes in the yeast genome, we can annotate each gene to a biological process. So that's really interesting in terms of this is the kind of work where we can use a systems biology approach and go towards functionally annotating every gene in the genome. So going back to chemical genetic interactions and where chemicals come into play. The idea is that you can do these screens, but in the context of a drug. And so what that means is analogous to what we have on the right with synthetic lethality. If you combine a drug that's inhibiting a target and combine it with a gene mutant, you can also read out those synthetic lethalities, and not just synthetic lethalities, but also quantifying the spectrum of chemical genetic interactions. And so that's what we see here with this profile, where instead of a query gene A knockout, we replace that with a drug and also profile that against a genome-wide deletion library. So what we have here are these chemical genetic screens. And if you think about these blue and yellow squares, they also represent sensitivity of a gene mutant to a drug or resistance of a gene mutant to a drug, respectively. And so what's really interesting is we can build these profiles and we can really integrate that from the left to the right. So integrating CGI profiles on the left with GI profiles on the right with the guilt by association principle, we can say something interesting about a drug here, that if it looks like a profile for gene A, we can predict that drug hits or targets gene A. And so this is really the basic principle for making drug mode of action predictions. Again, this Yeast work, which was done before my time in the lab, the idea was to take that Yeast genetic interaction map and then put compounds onto these bins in the map. And so this really becomes an approach to rapidly annotating large compound libraries and their mode of action in Yeast. And so this work was done back in 2017, published in Nature Chem Bio, where they took, so basically we took a compressed mutant genome collection. That is, we didn't take the entire genome-wide collection, but really compressed it down from 5,400 genes down to 300 or so diagnostic genes. And with this compressed profile, we could scale up these chemical screens in Yeast. And over 6 large compound libraries that were available, we could basically rapidly annotate those. And so of the 14,000 compounds in these 6 libraries, we made 1,500 high confidence predictions. So this is a really cool approach in Yeast. The question then is, can we translate this approach to human cells? And that is what my thesis work focuses on. So, you know, the SGA method in Yeast, we could do that with Yeast, and it's very facile and easy to make those double mutants relatively. How do we do that in human cells? The answer is we use these pooled CRISPR screen methods. And so I'll briefly describe what that method entails. The idea is that, say you can make a guide targeting each gene in the protein coding genome of for human cells. So there are 18,000 or so of these protein coding genes. We have multiple guides in that library targeting each gene. And then basically what we do is put this library into plasmids. And these are then packaged into lentivirus. And what you would do is infect a Cas9 expressing cell line. You then select for cells that have uptaken a guide RNA. And this is usually a puromycin antibiotic selection step, where that puromycin resistance cassette is built into the plasmid. So basically after this step, you have only selected cells that have uptaken a guide RNA. And you've done this in a way to ensure that only one guide is represented per cell. Now, in chemical screens, what you would do is then split this into a drug or compound arm versus a control arm. And you'll see as you let this experiment go out over a certain amount of time, some guides, like the blue ones here, would be enriched in the drug versus control condition, and some would be depleted. So at the end time point, you extract the genomic DNA from these cells, prep this for next generation sequencing. And the guide abundance or the read count data that you get back is essentially a proxy for cell viability. And so now that we have a proxy for the phenotype of interest, which is just fitness, we can then begin to build and quantify these chemical genetic interactions and build these profiles. And so just a little more about that approach and what we do computationally with our pipeline is that, again, we have our drug versus control arm, drug down here. And what happens is that there are going to be some guides that are more fit. They are enriched at the end versus the start of the experiment versus those that are less fit. And you can plot this distribution of fitness effects and then plot the fitness effect in compound versus control. And what you get is basically those that fall over here on the y equals x distribution are positive interactions, and those over here are negative interactions. And so from this, basically quantifying the distance from the y equals x line, you can quantify the CGIs and start to build these CGI profiles. So coming back to what my work tries to address is, first, we're trying to, if you remember back in the beginning, we're trying to come up with a more efficient way towards compound-centric drug discovery. Now, using this method, I described the Genome-Wide library for these pooled screens, which are over 70,000 guides hitting 18,000 genes. And that is really not a scalable method. The picture from colleague here, where performing these CRISPR screens takes a lot of cell culture reagents. And I have experience doing this myself. This is, if we want a good number of replicates for statistical power, this really can get out of hand pretty fast. And so the idea that we're borrowing from the Yeast work is, instead of taking the Genome-Wide library, can we choose the most informative gene deletions, that is the most informative columns, and compress this Genome-Wide library such that we have this targeted library, which will considerably cut down on the cell culture resources, will considerably cut down on the next generation sequencing costs, and thus make this a more scalable method. And so what I'm going to go into is the work that we've done on that front in building this scalable chemical screen platform. So this paper that Andy mentioned in the beginning has recently been released in START Protocols. So spent the past couple of years building a scalable CRISPR screen protocol in non-transformed human cells. This method could be used in multiple cell backgrounds, but we did want to point out a few things in our protocol that are different than other published CRISPR screens. And one is that so one advantage is that because of the smaller scale of these libraries and these screens, we're able to have more coverage. That means more depth representation of each library guide. So that's at 1000x, which is much more than the 250, 350x coverage from genome-wide screens. And also because we couldn't cut down on sequencing costs, we can actually sequence more intermediate time points. And so what I mean is that most published screens out there just take a T0 or T18 time point and just compare end versus start. But we're able to track the CGI data over time, over intermediate time points. And we're able to do that by sequencing it every 3 days. And I'll show in some later slides how biological and meaningful that could be. We also in this protocol provide some bioinformatics tools for quality control of these screens, as well as interpreting the data. In terms of these scalable screens, what I've done in conducted these experiments in Anya's lab was to do these 5 proof of principle screens. And so we chose these 5 compounds. So here, bortezomib, HU, camptothecin, olaparib, and colchicine, for 2 main reasons. One is that these are fairly well characterized. We know roughly their protein targets, as well as their general bioprocess targets in the cell. And secondly is that we have genome-wide screens to compare this data to. So in terms of we're doing these proof of principle scalable screens, we want to see if we can recapitulate the same signals or same CGI data. And so to that end, in terms of the library design, we're going from 18,000 genes down to 1,000. And we have 4 general categories in terms of selecting those CRISPR guides. First one here is, this is a DNA damage response biased library, partly because we're in a DDR lab and we are interested in studying these genes. The other categories here, we're looking for guides that capture the largest variance in the published CRISPR screen space. And we did so because we wanted guides that, that have variable phenotype across multiple screens. We also have a cluster here that can pick up subtle fitness defects over time, time course CRISPR screen data. And then finally, a category that basically includes genes that are frequent interactors. That is, that they have frequent genetic or chemical genetic interactions in CRISPR screens. So the scalable CRISPR screen library, again, 1,000 genes, and we had 3 guides per, so it was about a 3,000 guide library. Again, that is considerably smaller than the 70,000 guide library for genome-wide screens. So I'm showing here a global heat map of those 5 proof of principle screens. And here I just want to point out that this is not actually increasing dosage. This is increasing from left to right time points. But we're able to put all this data in one global heat map. And what I wanted to show is that we are recapitulating the expected CGI for these compounds. So here you can see a nice cluster for hydroxyurea, which we know interacts with the 911 complex. So you see those genes here. We also see for our genotoxins, camptothecin and olaparib, synthetic lethality with these homologous recombination genes. We see that here and here. And then finally here, this is a potentially novel finding, is that these chromatin remodelers here, this last cluster, has positive interactions with camptothecin, but negative interactions with colchicine. And this has, in terms of what we know, been previously unreported. So this is a novel finding from our chemical platform. What I want to do next is compare these scalable versus genome-wide screens. And so here are the table of parameters for each type of screen. So for the 5 compounds of scalable screens, we dose this at 20% inhibition, so 20% less viability compared to control. And you can see that we're sequencing this at many time points and higher coverage. I'll point out that for the genome-wide screens that we have to compare, the dosages are different. This IC20 is different in that for this Durachet screen set, we're determining this over 12 days instead of 3. For genome-wide screens done at Toronto, which is by our collaborators, they're hitting the cells at a much higher dosage. So keeping that in mind, the point I wanted to make is that these scalable screens have comparable signal to noise ratio. And so what I mean by that is, and I'll try to simplify this, is that when we look at these screens for HU, and here are the scalable screens that I had at T6, T12, and T18, and then the genome-wide screen for comparison. We come up with a gold standard set of genes that have CGI signal overlapping each of these time points in the genome-wide screen. And so that's the red dots. These are signals that we think are true chemical genetic interactions, whether they were found in our scalable screens or the genome-wide screens. And we're quantifying how far they are away from the noise cloud. And so the signal to noise ratio metric is just quantifying the average of all these distances away from the noise cloud for each of these red dots. And the point of that is to say, over time, well, first of all, the scalable screens all have comparable or higher signal to noise for the HU screen here. That's what we see with these 3 bars compared to this one. But also, interestingly enough, is that we see a peak in that signal at an intermediate time point versus the end time point that most people screen at. So we generally see this trend for, if I blow this up to multiple compounds, and for across all 5 compounds that we screened, we see a general trend where the last bar is the genome-wide screen. We see that we have comparable signal to noise, sometimes better than genome-wide, sometimes not as well, but at the very least comparable signal. And also that you see this general peak of the signal in intermediate time points relative to end time points. So all that to say that these scalable screens are having comparable signal to noise ratio. We're collecting similar functional information, yet we're doing that with a compressed library. And that's what makes this method scalable. So in terms of biological interpretation of this data and what are some potential novel findings. One is if we look at just each DNA repair pathway. So here on the left, I'm looking at HR genes. And so I subsetted that gene set out from the global heat map. And you can see that I'm going to focus on camptothecin here. We have these expected negative interactions with HR genes. And so it's good that our platform is picking that up. Now, on the right, what's a new finding is that we have interstrand cross-linking genes here. So this repair pathway where the major players here are the FINK genes. And what's really cool is that you can see this temporal resolution of these FINK gene cluster lighting up at the end time point in response to camptothecin. And so that has not been described in the literature before. We suspect that there is this pathway switch, or at least an activation of this ICL repair pathway in response to camptothecin that has not been seen before. So this is just showcasing that with this temporal resolution, this intermediate time point data, that we can pick up some new biology. So other than temporal resolution, we can also differentially cluster different players in DNA replication. And so what you see here is when I subset out the DNA replication genes on the rows, that we can get a nice separation of lagging strand, so here on the bottom, and leading strand genes in these two clusters. And so the thinking here is we can pick this up with camptothecin because if you knock out these lagging strand players, that's highlighted in yellow, representing these positive interactions, they're basically surviving. A positive interaction means that they survive better than in control conditions. And so what that means is, or what we suspect is that, well, you're knocking out players that's going to delay Okazaki fragment maturation. You're going to slow replication. And why is that happening? Is that when you have camptothecin, that compound is going to get stuck on the DNA and create these adducts. And when you have slowed replication by knocking out these genes, you're going to see and meet less of those adducts. And so the repliposome machinery will slow down, see less of these adducts, and actually survive better as a cell. Whereas you see the opposite effect of where you're knocking leading strand genes out, you will essentially just kill the cells. These are just essential. So it's really cool to see that our scalable platform is picking up this kind of differential biology. And the last point I want to make with these proof of principle screens is that when we look at their chemical genetic interaction profiles, we ask whether those interaction profiles are enriched in the mode of action of the compound. And so our approach here was, we know the general targets of each of the 5 proof of principle compounds, and we can associate a biological process term, a proxy for their mode of action, and mine the genes in the scalable library that are annotated to that mode of action. And so all this, when we combine all this data, what we're saying here is that we have statistical significance in picking up the mode of action for each compound across, if you look at the entire genetic interaction profile, or if you split it into just the negative side and the positive side, you get that significance on the negative side, but it's also trending in the same direction with the positive side. So all this is to say that the chemical genetic interactions can tell you something about the compound mode of action. So just to summarize this section, we've developed a scalable approach to chemical screens at 1/20th the resource cost, cutting down on the amount of cell culture plates and the next generation sequencing costs. And what's really cool is that with this compressed library approach, we get comparable CGI signal compared to the genome-wide screens. And thus this is lending evidence or credence towards we can use this approach in a scalable method with this compressed library. And then finally, we get some interesting biology when we look at the temporal resolution of the CGI data, specifically here displaying the timing of the repair pathways. Okay, so I'll move to the final section of the talk, which is focusing on the mode of action predictions. And so we use this computational tool that's been developed in the Myers lab called CG Target. And I'll go into what that is. So there's 2 modes to CG Target, both trying to answer the question of, can we predict drug mode of action from CGI profiles? And so on the left, mode A is looking at just the profile, the CGI profile itself, looking at the positive or the negative ends, and just doing a direct enrichment on those top hits from either side. So that'll give you a sense of, which genes are the strongest CGI hits. Are they pointing to biological process or pathway terms that hint at the compound mode of action? But on the right is going back to the background for this talk. If we integrate those profiles, those CGI profiles with a genetic interaction network or genetic interaction profiles for human cells, then we can begin to think about mapping these compounds onto the genetic interaction map. And so to go into that in more detail, this was the CG Target work done in Yeast. And there's 2 components to that mode B approach. So the first one here being that you take a CGI profile and you can correlate this to every genetic interaction profile in Yeast. So this right here I'm showing in Yeast. And then we can, using a correlation metric, rank the profile similarity such that you're coming up with a rank of genetic targets between the compound and each of these query genes on the row side. Now, so that's the compound to gene mapping. For gene to bioprocess mapping, what we do is feed in something like Go biological process and aggregate these profile similarity scores into and bin them into bioprocesses. And so when we do that, you can then rank these process level predictions. And the top ranking, say process A here, would be the mode of action prediction for that compound. So in essence, we're basically mapping compounds to biological processes. Now, in terms of this, so that was all done in Yeast. And again, with the SGA method, it's been easier to construct these screens. In human cells, it's a little more laborious. And so I will describe here the, so in terms of, we need those GI profiles in human cells and that GI network to begin implementing this method. And so what we've done is that in collaboration with Charlie Boone and Jason Moffat over at the University of Toronto, we have begun constructing that interaction map in human cells. And we do this in the context of these near haploid hap1 cells that are a derivative of a CML. And we've completed 200 queries. So you can imagine for that genetic interaction matrix, we have 200 rows completed against the Genome-Wide library. So you can appreciate the scale, the space here. We have 20,000 protein coding genes, and we're only hitting 200. But what we found is that the initial 200 set still informative for building a first draft of the human genetic interaction network. And so that's what you see here, is that from those 200 query screens, we can already build that first draft of the network. And this is already self-organizing in terms of we have up to, let's see, 17 different biological process bins here for human cells. And so I'm actually going to go back here. So the idea is that, so we have these 200 or so queries, 200 rows here. We want to also build out the analogous CGI profiles in the same cell line background and same context. And so the same collaborators have begun doing this, and they've screened over 100 compounds, I believe 135 unique screens. So 135 of these rows on the left already built out. And so if we have the GI profiles, the CGI profiles, and the mapping of these genes to biological process, we can start to put these compounds on the map. And so before I show that, I just want to show what these results generally look like. And I'm using a scalable Portismo screen for comparison. The first kind of table or result that you get is these gene target predictions. And so that's going back to those profile similarity scores. And for the bortezomib compound, we can rank the gene targets by that similarity score. And so what I do want to show here, point out here, is that PSMB2, a proteasomal subunit, comes up to the top of this list, this very lengthy list. And that's promising. Bortezomib is a proteasome inhibitor, and we would expect these proteasomal subunits to come up to the top. What's also interesting is that there are some genes here marked in red that have to do with the nervous system. And then I'm actually going to go into the next table, or the second kind of results, which is the bioprocess target predictions. And again, that's aggregating the gene target scores that we saw in the previous slide into bioprocesses, and then ranking those. And so what you see here is that these bioprocess prediction tables do better. We're not looking just gene to gene anymore. We're looking at clusters of genes. And you'll see within the top 4 rankings, these proteasomal subunits come up. And we usually associate bortezomib or annotate it with this particular Go term right here. So that's basically saying that our scalable screens work, our computational prediction tool works. What's particularly interesting about this set of results is that we know that bortezomib has the side effect of peripheral neuropathy. In fact, this precludes its clinical use for many patients. And what I marked in red here is basically the off-target or side effect of bortezomib in terms of all these nervous system GO-BPs coming up. And so this is just to point out that with our scalable platform, with this computational tool, we may be able to pick up not only the primary mode of action, but secondary or off-target effects. So then back to the idea of placing these compounds on the genetic interaction map. I have the human genetic interaction map on the left here. And this is some preliminary data where we looked at genome-wide screens for a couple of compounds. Again, all done at the University of Toronto. And start to bin these compounds on this map. And so the idea here is that we see enrichment of the negative CGIs for the bortezomib profile in red here. And it lights up in a few of these functional bins. But what's interesting is that it lights up here specifically. And this cluster here is pointing to protein degradation, which is exactly where we would expect bortezomib to be. So that's this yellow cluster right here. Oh, looking at a couple of other compounds, camptothecin, which we know targets DNA repair and replication, conveniently lights up only in that bin here. So this is the DNA replication repair bin in yellow here. And we see that nicely be enriched over on this side. And then finally, we see this with colchicine as well. So this blue cluster of mitosis and tubulin cytoskeleton, we see enrichment of the negative CGIs for colchicine light up in that area. So this is all to confirm that our platform is working, our computational predictions are working as expected. We are able to recapitulate the mode of action for these compounds. And so you can imagine if we were to scale this over large compound libraries, we could essentially characterize compounds where we don't, the mode of action is previously uncharacterized. We can place those on the map. That can point us to understanding the drug mode of action and developing that as a therapeutic. So just to summarize this portion of the talk, this computational tool in CG-Target, which was previously used for the yeast work, I spent my thesis work, my time in the lab, adapting this tool for the human cells and making mode of action predictions for compounds. In terms of looking at scalable versus genome-wide screens and the application of the tool on these screens, I haven't shown data for this, but preliminary results show that the integration approach, so mode B, is outperforming just the baseline mode A approach for scalable screens. And so that's interesting because I spent the first part of the talk talking about the scalable screen platform. If we do that at scale with the compressed library and with lower resource costs, we can still capture the mode of action using this integration of profile method. Now, when we look at genome-wide screens, there's complementary predictions from both mode A and mode B, which kind of makes sense that with mode A, you have an entire profile to enrich on, and you can make pretty good mode of action predictions just off of the top positive and negative CGI hits. But still, we found that mode B still complements those mode of action predictions and sort of adds information on its own. So again, we've made these mode of action predictions with using CG Target for 135 genome-wide compound screens over Toronto. And we're looking to, in terms of our collaborative efforts there, we are performing many more compound screens and hoping to make more mode of action predictions. So that's pretty much it for my talk. I wanted to just summarize the takeaways here. So one is, in describing the chemical screen platform, this is a scalable approach to compound-centric drug discovery. And we're doing this at 1/20th the resource cost, with cell culture reagents and next generation sequencing costs being drastically reduced. This is an approach for annotating large compound libraries. So like what we saw in yeast, we could apply this to human cells as well. And potentially a powerful approach for exploring uncharacterized compounds or repurposing drugs. And then just some final thoughts from my end. I've been fortunate enough to sit at the intersection here of the bioinformatics, the basic biology research, doing both wet lab and dry lab, and then also understanding, putting on my clinical hat and understanding the application of this work. So it's been a really unique experience from my end, seeing how all three fields intersect. And just in terms of some final clinical thoughts is the impact on drug discovery. Like I mentioned at the beginning of my talk, the idea is to make the drug discovery pipeline more efficient. And this, what I presented today is potentially a step towards that. So yeah, I think I have some time left. So I just wanted to acknowledge both Anya and Chad's labs, as well as my thesis committee, which also includes Zohar Sachs and Stephanie Huang. Also, want to acknowledge the MSDP for their support through all the years of training, as well as the TL1 and F30 grants from NIH. So yeah, with that, I believe I have about 5 minutes left. I'd be happy to take any questions.
Kevin, thank you very much. I appreciate it. I learned a lot. Yeah, with all things bioinformatics, I always tend to kind of get amazed by the plots, and then kind of trying to figure out how to ask detailed questions is a challenge. I have a couple, but I see Scott came on. So Scott, I'm going to turf it to you first, sure, and then I'll put mine in later.
Yeah, Kevin, that was really cool. I was wondering, with the chemical screening that you've looked at, if you've pulled out any of the kind of pen assay interfering compounds or the dirty drugs that are known to hit multiple targets. Curcumin comes to mind, resveratrol comes to mind. I work in the prostate cancer field, and these are drugs that have been reported to inhibit androgen receptors, but also inhibit many other things too. And so if you project these kind of dirty drugs against your genetic interaction maps, what do you see?
So I don't think we screened those compounds in particular, the ones that you mentioned. We have some pan kinase inhibitors as part of our screens, and those tend to, they have many targets, and those tend to be difficult to work with. The data is, we do see in terms of preliminary results that they are hitting various bins in the map. I would have to go back and look at those more closely. But yeah, I think what you brought up is a good point, that drugs that are, dirty drugs in terms of they hit many targets. It is sometimes difficult to interpret their data. We know also that the compound dosage affects, say, the selectivity of the targets that they hit. So that's something to keep in mind, like the drug dosage really impacts the data from these compound screens. But yeah, that was a good question, good point.
Cool. Any other questions before I jump in? So one of the things, Kevin, that I wanted to ask, and I apologize if it's naive, and I apologize if I happen to miss a subtle but important point kind of in the early introduction. With these CRISPR screens, with the guide RNA libraries that you're using, are these predominantly, and this is my assumption, predominantly generating loss of functions across the genome with these targeted CRISPR screens, or did you have the opportunity to also introduce potential gain of functions, mutations? That the libraries can be screened, the chemical libraries can be screened for those interactions as well.
Yeah, so these are loss of function. So these are complete gene knockouts. These. So what I presented is just gene knockouts, but there are other efforts that use like CRISPR-I, CRISPR-A, where you can mimic like inhibition instead of total knockout or activation of genes. And there are other methods out there that also do the gain of function mutations. So this is certainly possible with
it gets challenging. It gets challenging quick. And then I also have another question that is kind of mundane and rote and technical, but at the same time too kind of important. All of us that use CRISPR in our labs to do various things, to create genetic models, CRISPR has off-target effects. Obviously, you have statistical replicates throughout your experiments in order to moderate that or control for that. Is there anything else about the genetic analyses? Is there anything else about the quantitation, the sequencing that can help to also get a sense of if any of the CRISPR screens are generating off-target effects, or just relying on good old-fashioned have enough replicates to mediate that?
Yeah. So having enough replicates is important. That's we actually do. So we perform all those screens in triplicates. And if you think about, we have, at least in the scalable screens, we have 3 guides per gene. The genome-wide, we have 4 guides per gene. And so you're essentially getting like, triplicates times the number of guides, you get 9 to 12 readouts per gene. And so that kind of controls for off-target things. Also, in our computational pipeline, we identified or flag guides that are sort of potentially outliers. They're not in concordance with the other guides. And also just in terms of the construction of these libraries, both for the scalable and genome-wide libraries, that there has been careful design and selection of the guides that are quote unquote best performing, that they do knock out the genes as expected. So
sure.
Yeah.
Cool. Thank you.
Yep.
Well, any other questions then before we break? All right. Going once, going twice, sold. So thank you again, Kevin. Appreciate it. It was great to actually get a chance to see all of your work. I know we've talked about it over the computer and the microscope doing sign out. So it's fun to see everything you've been working on. So appreciate it. Thank you so much.
Yeah, for sure. And thank you for having me.